Hi, I'm Ali Torkamani, an assistant professor and computational biologist here at Scripps Translational Science Institute and the Scripps Research Institute. My role at STSI and TSRI is twofold. One of the things I work on is just providing bioinformatic and computational support to a wide variety of investigators at TSRI. So this can involve both help with design of projects at the sort of upstream stage or analysis of data at downstream stages. So an example would be primer design for bisulfite converted DNA, where you're trying to do target capture of a large number of genes within the genome by PCR of bisulfite converted DNA. So I would write an algorithm that sort of targets the regions of interest and provides a list of primers that can be used to capture those regions. And I do all sorts of other sequence analysis of various types, mostly revolving around next generation sequencing data, but also including other types of genomic data, especially expression data or epigenetic data. My own personal research interests are in the realms of network analyses, pathway analyses, and also genome annotations. And these two things are sort of interrelated with one another. So on the network analyses side, instead of doing genomic studies where you're looking at one gene at a time or one mutation at a time, I use network-based approaches or pathway-based approaches to combine genes and mutations together and look at the combined contribution of genes or mutations across specific networks or pathways. And these networks or pathways have genes within them that are related to one another based on their role in similar biological processes. It's a data reduction technique, essentially, that increases your statistical power, since generally in studies you're looking at 20,000 plus genes, possibly millions of mutations. So just in a statistical sense, it's hard to get enough statistical power to separate noise from signal when you're looking at that many signals at once. On the annotation side of things, in a similar way, take mutations from sequencing studies and try to take what is approximately 3 million variants per genome and reduce that to a smaller set of variants that are predicted to have some functional impact. So we take variants, see where they lie in the genome, determine how they affect genes, whether or not they're in promoters or within the coding region or within introns and may affect splicing, then apply a series of prediction tools to determine which one of the mutations within the promoter may actually affect transcription, for example, versus other mutations within the promoter which may just be neutral. Prioritize that set, bring it down to a smaller set, and then perform statistical analyses on that. And the network approach fits in here as well, since you can prioritize your variants and then start asking questions about sets of variants that are perturbing protein function across a large number of different genes who are related to one another based on the network or pathway that they participate in. So in that way, I try to do a lot of data reduction, data cleaning, sort of providing network-based and computational insights into variants, and hopefully at the end of the day we can increase the power to detect signals and determine which genetic variants are involved in various human diseases.